we have red blood cells. These red blood cells are red in color due to presence of hemoglobin, that you know. That determines ABO blood group. Why? Because ABO blood grouping are controlled by the gene I. Gene I controls the ABO blood grouping. Okay. This gene I, plasma membrane of red blood cells has, if you take the RBC, okay, let us take the RBC. The plasma membrane of RBC, the membrane that surrounds the RBC. Okay. The plasma membrane of RBC contain, okay, specific sugar polymers. What are they? Sugar polymers that protrude from its surface. Okay. And the kind of sugar is controlled by the gene I. Okay. The gene I has 3 alleles. If you take the gene I, it produces 3 alleles. What are they? Capital IA, capital IB, and small I. Small I is completely recessive in nature. Why? Because it doesn't produce any sugar polymers. Capital IA, capital B are dominant in position. Okay? Because humans are diploid cells. Humans body made up of many diploid cells contain 2n chromosomes, which means 23 pairs of chromosomes. Okay? Okay? Any 2 of 3 I gene alleles, if you carefully see, and whereas if you see IA, comma, capital IB completely dominate over small I. In other words, IA and small I are not only present, and IA expresses, because small I does not produce any sugar. It does not produce any sugar. Doesn't produce any sugar. When IB and small I are present, IB express. What is expressing? IB express. Now in case IA comma IB are present together, IA comma IB present together, this produces together their own type of sugars. This is because IA, IB both can be dominant together, hence it is called as a codominance. This codominance, hence red blood cells are both A comma B, both sugars which are dominating in position. So hence there are 3 different alleles. How many are there? 3 different alleles which forms 6 different combinations of genotypes. What are those 6 different types of genotypes? I would like to explain for ABO blood group pattern in this table. Let us take capital IA from parent 1 and capital IB from parent 2, and the possible genotype will be IA comma IB, IA comma IB, and the possible blood group will be AB. In the similar manner, from parent 1 IA allele is there, from parent 2, and if you see IA allele is also there. Possible genotype IA comma IA, and the blood group will be A. Okay? IA allele is from parent 1, small I is from parent 2, IA comma small I, genotype is there. Possible blood group will be A. IB allele from parent 1, okay, small I from the parent 2, and the genotype will be IA comma small I, IB comma small I, okay, which resulted in a B group blood. From parent 1 IB allele is coming, from parent 2 also IB allele is coming, IB comma IB, possible blood group will be B. Okay, and whereas from parent 1 small I is there, from parent 2 small I is there. Okay, genotype will be small I, possible blood group will be O. Why? Because if you take the small I, both are recessive in nature, which resulted in the formation of new blood group called O. Why? Because small I will not produce any sugar polymers, hence they become recessive for O group bladder. And this is about a codominancy. A single gene product may produce, okay, if you take a single gene product produce more than one effect. Okay, let us take the starch synthesis in a pea seeds. Is controlled by one gene. It has two alleles. Okay, how many alleles are there? It has two alleles, capital G, small p. Starch is synthesized effectively by capital B, homozygotes. What are they? Homozygotes. Large sized starch grains are produced by capital B, genotype homozygotes. In contrast, small p homozygotes are there. These small b homozygotes have lesser efficiency in starch synthesis and produce small starch grains. Whereas finally, if you see, after maturation of the seeds, capital B seeds are round in shape, okay, and the small p are wrinkled in shape, okay. Heterozygotes produce round seeds, but if you check heterozygotes that are coming from these plants, they are round in sheets, and so capital B seems to be dominant allele, as you know that capital B for the starch synthesis is dominant allele, whereas small b is recessive in nature. Okay, so in this case, starch grain size is considered as a phenotype. Starch grain size is considered as a phenotype allele. Okay, remaining part we will continue in the next class, until stay tuned.